All right, I think we are ready for the last talk of our second session, and we have our guest speaker. It is a tradition of the Geroscience Symposium at the University of Washington to have guest speakers from other aging training programs around the country. And Kenny Wilson is joining us from the newly funded Buck Institute and University of Southern California Nathan Shucks Center, but I think actually the training grant has been going on for a little bit longer. Well, Kenny, take it away, and you're going to talk to us about OXR1 and dietary restriction.
Do the slides look okay?
They look great.
All right, great. Thank you, everyone, for having me here. I'm excited to talk to you about this protein that we've been working on called oxidation resistance 1, and some of the work that we've been doing in finding a new pathway that's involved in. And I've been doing this work in the labs of Lisa Ellerby and Pankaj Kapahi. To start off, we started looking into dietary restriction and trying to figure out what's going on, how that is known to extend lifespan and health span. I won't go into too many details because a lot of it's been talked about in the first section, especially. But essentially, we wanted to figure out what are some new mechanisms that regulate dietary restriction in addition to the ones that are already well studied. And the reason that we really got into this was because of this paper that came out about 9 years ago, in which they took different strains of mice and put them on two different diets, one that's an ad libitum condition and one that's a calorically restricted condition. And they found that the different strains actually responded differently to dietary restriction. Some of them actually even had a detrimental effect. Like you can see this strain here, 115, lived far shorter under dietary restriction than it did under an ad libitum condition. And so we were curious in understanding what could actually be regulating these responses. Why does the genetic background influence it? And so what we used for this was the Drosophila genetic reference panel. It's about 150 to 200 strains of flies. They're all genetically distinct. They were originally caught at a farmer's market, actually, and then inbred. And then they were sequenced. And so they know all of the different genetic changes to all of these different strains. And so what we did is we took all of these different strains, put them onto two different dietary conditions. One that's dietary restriction, which had 0.5% yeast extract, which is their protein source. And so in this case, it'd be protein restriction. Or an ad libitum condition, which is a tenfold increase in the amount of yeast that they are getting in their diet. And we've tested a number of phenotypes for this. The first publication that came out of this was about 4 years ago. We tested a number of different traits associated with metabolism. So body mass, starvation resistance, et cetera. And this year, actually, we've had a few publications come out regarding this. One in which we measured the length of life and how long these animals were actually able to stay active from dietary restriction. A recent paper that came out with a collaboration with the Promislo Lab at Washington, and Kelly Jin led this. She'll be talking later. And then a recent paper that was also accepted studying how post-irradiation stress in the intestine is affected by genetic background. And today I'm mostly going to be talking about this first one that came out earlier this year, lifespan and health span. What you're seeing here is all of those different strains in the Drosophila genetic reference panel arranged according to how long they lived on average. And this is 200 strains that we tested per diet, per strain. And so what you see is that there's a big variation in the amount of and how long these flies actually live. So on ad libitum condition in the red bars, and then dietary restriction is the same strain, or the blue is the same strain, how long they lived on dietary restriction right next to those red bars. So you can see on ad libitum conditions, some strains didn't even live a week. Some lived up to 50 days. On dietary restriction, in general, it was beneficial. But you can see there's a number of longer lived strains on ad libitum conditions that actually lived shorter with dietary restriction. This is consistent with the idea that DR is not beneficial for everyone. And then we also tested as a form of trying to measure how healthy they are, how active they are, and how well they can climb their vial that they're in. And again, you see dietary restriction was predominantly beneficial for all of these. And through doing genome-wide association studies, we were able to find a few genes that regulated each of these phenotypes. One of the ones that regulated lifespan we found, and we called it decima. It's involved in insulin-like signaling. In particular, it's involved in the production of insulin-like peptides. And then we found this gene Daedalus, which actually, when we knocked down Daedalus, these flies became more active, but only under dietary restriction. But today I want to talk about another protein that we found, another gene called oxidation resistance 1, which regulated lifespan under dietary restriction specifically. And here's what we found when we lined all of these different strains, each dot representing a single strain from the Drosophila genetic reference panel, aligned them according to their phenotype, in this case, median lifespan rather. And it's separated according to their genotype. So if they had a G at this locus, they're in this column here. If they had a C, they're in this column here. And then this part here is ad libitum diet. On the right here is dietary restriction conditions. So you can see the black bars, which represent the average, there's no difference in ad libitum conditions, regardless of which genotype these flies had. But in dietary restriction, you can see that there's a shift where the strains that had the G at that locus actually on average lived longer than those that had the C. So we were kind of intrigued about what could be happening with this particular protein. So we asked, what is known? What is already known about oxidation resistance 1? Well, it was originally discovered to protect against oxidative stress. That's why it's called oxidation resistance 1. That came out about 20 years ago. Well, in more recent years, it was found that actually overexpressing oxidation resistance 1 in an ALS model, a SOD2 knockout model, overexpressing OXR1 actually rescued the phenotypes that they saw and the shortened lifespan in particular. More recently, a paper that came out of the Bellin lab in Houston, they found that there was a genetic variant in humans that actually causes an undiagnosed disease in these humans. And actually the people who have a homozygous mutation for this gene don't make it past their teen years. They die very early and they have a very strong degenerative phenotype. And actually reducing OXR1 activity caused lysosomal activity, overactivity in both flies that they tested it in, as well as the fibroblasts that they took from the human patients. And actually this was independent of oxidative stress. So OXR1 seems to have a few roles that aren't necessarily restricted to oxidative stress in particular. And then more recently with some collaborators, there was a proteomic analysis that was done that saw that OXR1, when it's down in humans, is highly correlated with Alzheimer's disease. And I'll actually show that later in my presentation. But first we wanted to know how it influences lifespan. So we did an RNAi screen for all of our different candidates, but I'll show you here OXR1's results. So this is inducible pan-neuronal knockdown of OXR1, and this is only in adult flies. And when we knocked it down under dietary restriction, again shown in blue, the solid lines being the control condition, the dotted lines being the knockdown. On dietary restriction, there was a shortening of lifespan. And it actually almost looked as close as the lifespan I had on ad libitum conditions. But interestingly, knockdown on ad libitum conditions did not have an effect on lifespan. And so we thought, okay, what happens if you do a non-inducible knockdown of this, including in development? And interestingly, these flies, very few of them, first of all, made it to adulthood. But the ones that did died in about 10 days. And you can see, especially in dietary restriction, when they lived out to about 75 days, it's a massive reduction in lifespan. And also, interestingly, though, dietary restriction didn't benefit these flies at all. There was no extension. And so we thought, what could a genetic variant in particular could be doing to this gene? And so we looked back at the Drosophila genetic reference panel strains. We took a few strains that had the short-lived allele and a few that had the long-lived allele, raised them on dietary restriction for a few days, and then measured the expression of OXR1. And interestingly, in the long-lived one, you see that there's a huge increase in the expression of OXR1 on dietary restriction. But the ones that were short-lived did not have that increase. In fact, in most cases, they had reduced expression of OXR1 under dietary restriction. And we wondered what happens if you actually overexpress this protein then. And we saw that an overexpression of human OXR1 in these flies actually extended lifespan only under dietary restriction. So there's some interaction happening here that's specific to dietary restriction that's regulating lifespan. To figure a little more about what's actually happening with OXR1, we did a RNA sequencing analysis where we knocked down OXR1 in some flies, raised them on a couple diets, the 2 diets I've talked about, and took the heads and checked the transcriptome at different days, different time points in these flies. And I want to focus on one in particular, the ones that were showing a knockdown of OXR1 on DR at day 14. The Go terms associated with these genes that were knocked down as well involve rhabdomere formation in the eye, ENA-D signaling, which also senses light in the eye, response to external stimuli, detection of stimuli, cell projection. All of these things had to do with the eye development of the fly. And so we decided to look at the eye, of course. Now, normally a fly eye looks very orderly like this here, and you see there's 7 photoreceptors in each of these ommatidia of the fly. But when we knock down OXR1, you can see it's in complete disarray. There's a huge amounts of cell death, and essentially there's just no order whatsoever to the fly eye. And when we tested how well these flies can respond to light here in this phototaxis assay, we took the flies, put them in a long tube and shined a light at one end. Typically they go to it. You all know mosquitoes like to go to a mosquito light. We did the same thing with the flies. On dietary restriction, you typically see this big increase in how well the flies respond. But with the knockdown of OXR1, it essentially didn't happen. There was no increase over that of an ad libitum diet, and actually they had just had reduced levels overall throughout their life. So we wanted to look a little bit more about what was happening morphologically to these eyes, and we looked back at that paper I mentioned earlier by the Bellin lab. And this is a figure from that paper, what they showed. So in a control, this is the R7 photoreceptor of the fly eye. In your control, it looks fairly normal. When they show the RNAi for OXR1, you see that the photoreceptor as a whole is getting dark, and you see these big black spots that are showing up. And those are lysosomes. I mentioned earlier that they showed overactive lysosomal activity. Essentially, they're turning black because they're so stressed. And it reminded us of another paper by the same lab, in which they saw in the photoreceptors of flies as well, these black accumulations, again, lysosomes. And they linked it to something called the retromer. And so to give you a little background on what that even is, when a transmembrane protein is endocytosed, it typically has two fates. One is it can be sent to the lysosome for degradation, or it can be recycled. They call it the recycling pathway, but it's more like a reuse, because if you think of recycling, it's typically breaking something down like autophagy. But reuse is another thing that can be used a lot. And the complex of proteins that's responsible for this reuse pathway is called the retromer complex. And there's a few key proteins I'll point out here, VPS35 being one of them. Sorting nexins are another key proteins that are involved, VPS29, VPS26. We decided to look into this a little more, but the key thing about this is if you knock down the retromer activity, everything goes to the lysosomes. And that is what we were hypothesizing is why we were seeing these black stressed accumulation of lysosomes. And so in looking at how the retromer is changed with OXR1, we took our fly model, which knocks down OXR1, looked at VPS35 and VPS26, and we saw that there was reduced protein levels of these particular proteins with OXR1 RNAi. And in a mouse model where we overexpressed OXR1, we saw other proteins that are involved in this process, sorting nexin 1 and RAB7. Those are increased with OXR1 overexpression. And so we hypothesized that there's some sort of interaction potentially here between OXR1 and the retromer complex. And we wanted to figure out more of what could be happening there. But more so than that, we wanted to know if stabilizing the retromer complex as a whole could rescue this effect we're seeing with OXR1 knockdown. And so here's, again, what happens when you knock down OXR1 with your controls in the solid lines, blue R being DR and red AL, and then your knockdown of OXR1. When we overexpressed VPS35, one of those retromer components, we saw a complete rescue of lifespan and an extension of lifespan on dietary restriction, similar to what you see in normal conditions. We wanted to test this another way, and we found that there is a pharmacological tool that can be useful. R55 is a chaperone molecule that binds between VPS29 and VPS35 and strengthens their bond. And so we thought if we could strengthen this, will we get the same effect as we get with the overexpression of a retromer protein? And so again, here's your control conditions with your knockdown of OXR1. And with 6 micromolar R55 in the diet, we again saw an increase in lifespan and a rescue of the dietary restriction extension. And actually, they live longer than controlled dietary restriction flies do, suggesting that R55 could be really useful, especially under dietary restriction. Now to kind of shift a little bit more into how this influences diseases, I mentioned earlier that there was a proteomic analysis that took post-mortem human tissue and analyzed the proteomes of all of them to figure out any sort of correlations between Alzheimer's disease and these proteomes. And what we asked them to do is look at OXR1. And what was found is that in all of these different conditions, here's your, on the left is your controls, the asymptomatic Alzheimer's disease in the middle bar, or the middle box here, and then those that were diagnosed with Alzheimer's disease. The people that had below average abundance of OXR1 had all been diagnosed with Alzheimer's disease. And that was true in a couple of different tests as well. The CIRAD score for behavioral testing in Alzheimer's disease. Again, everyone who had reduced OXR1 also had the worst score imaginable for this possible test. And for Braak score for pathology as well. Again, lower OXR1 equated to reduced signaling of, or a reduced abundance of OXR1. And so we wanted to test how OXR1 could be used for Alzheimer's disease. We took a control fly, and what you can do with these flies is you can overexpress a particular protein involved in a disease like Alzheimer's disease, and then put it in the eye. So here we overexpressed human mutant tau, and what we saw is, again, a rough eye phenotype. You see these black spots. The eye doesn't look nearly as smooth as it does normally. But when we overexpressed OXR1, we saw a return back to this smooth eye phenotype. And it was essentially overexpression of OXR1 was negating this effect we saw with the mutant tau. And so our working model right now, we know that DR is somehow driving this function of OXR1, and that OXR1 is functioning in collaboration with the retromer complex. We're not quite sure how, but the important thing is that this recycling process seems to be really important for the mechanism here. And so with that, I'd like to thank a number of different people. Of course, Lisa Ellerby for hosting me in her lab now and teaching me a lot about neuronal biology. Pankaj Kapahi, where I did my PhD in his lab, and all the people who helped collaborate with this project as well, Rachel Brem, the Bellin Lab, and collaborators at Emory University. And with that, I'd like to leave you with this message that I'm passing around, that it's important to recycle. And with that, I'm open to any questions you have.
Fantastic. Thank you so much. Beautiful talk. All right, we have already one question from Shane Rea. He'd like to know where the fly SNP in OXR1 sit, whether it is exonic, regulatory, or intronic.
It lies in a region that is upstream of a few of the transcripts of OXR1, not all of them. It's not in an exonic region. And so actually we started looking at different proteins that could be binding to that region, and we found a few transcription factors that bind there. So we're actually starting to look into that. We found one that we think is a repressor, one that's a promoter of OXR1.
Right. And Mitchell, you have a question too?
Yeah, Kenny, great talk.
Thank you.
So you have this remarkable eye phenotype that you see with the OXRs and with tau. I know that in a lot of cases that can be used as a really effective readout for just sort of like general neuronal dysfunction. Mm-hmm. Are you thinking these mechanisms really kind of relate to something indirect of the eye, or that the eye itself is really involved in this?
Yeah, I didn't talk about that too much, but that other paper that came out from the Bellin lab, they actually looked at the whole brain of OXR1 knockdown flies and saw that the brain as a whole is messed up, lots of holes everywhere. And they mentioned specifically the R7 photoreceptor in the eye. That is the photoreceptor that actually descends into the brain. And so what the hypothesis is the brain is degenerating, and that's kind of giving a signal to the eye, which is degenerating as well.
Okay, Timothy Mackey is asking, do you see changes in endocytosis or autophagy in OXR1 knockdown, since those are upstream of retromer sorting?
Mm-hmm. Good question. That's exactly what we're looking into right now. As we speak right now, I have a Western blot going to figure out how LC3 levels are changing in these, so I can figure out lysosomal activity. And I'm also going to be looking at endosomal endocytosis in general.
Okay, fantastic. I don't see any more questions, so I think we can break for lunch 1 minute early. I would urge all the speakers to go back to their Slack channels, and I think there's 3 questions unaddressed